Hi, everyone, and welcome to today's webinar, Revealing 3D Structure of Nanoparticles with Cryo-electron Tomography, presented by Dr. Eric Bushong. We're happy you're here. We'll be reviewing how cryotomography revealed the true 3D architecture of complex nanoparticles. You'll see real examples of LNPs and viral particles reconstructed at nanometer resolution. You'll learn to identify structural defects that standard methods miss and discover how 3D visualization can validate your particle design and characterize heterogeneity in your samples. Before we begin, let's go over a few housekeeping notes. The webinar will be followed by a short Q&A session. You can submit your questions at any time in the Q&A box in the toolbar below, and Eric will answer them following the webinar. Any questions we don't get to will be followed up via email. Now, it's my pleasure to introduce our speaker, Principal Scientist at NIS, Dr. Eric Bushong. Prior to joining NIS in 2021, he spent 15 years working as the project scientist at the National Center for Microscopy and Imaging Research at the University of California, San Diego. Eric has a PhD in biomedical sciences from UCSD and has extensive experience with transmission electron microscopy, scanning electron microscopy, micro CT imaging, and light microscopy. Now, please join me in welcoming Eric. Eric, over to you.
All right, today I'll be discussing cryo-electron tomography and its application to the study of nanoparticles. This presentation is not going to presume any familiarity with tomography as a technique or even electron microscopy. I'll begin by reviewing why cryo-electron microscopy is a powerful technique for looking at nanoparticles and drug formulations. And then I'll discuss the motivation behind using cryo-electron tomography and discuss how that process works. And then finally, I'll present a few examples of nanoparticles that were imaged using cryo-electron tomography and demonstrate some of the insights that can be obtained using tomography. I'll briefly describe who we are. NanoImaging Services is a CRO that's been providing cryo-electron microscopy imaging for 19 years. The company started as a nanoparticle characterization service, and then in 2017 we began providing protein structure determination. And last year, Proteus joined NIS, adding protein production to our portfolio, allowing us to provide a complete path from gene to structure. And we're registered with the FDA as a GMP facility, allowing us to deliver GMP-compliant nanoparticle characterization studies. We do have a lot of experience using electron microscopy to characterize many different sample types, including VLPs and viruses, iron nanoparticles, liposomes, and lipid nanoparticle formulations. Before cryo-electron microscopy was developed, the traditional method for preparing nanoparticle formulations for electron microscopy would be to deposit the samples on a small grid with a continuous support film, and then allow the sample to dry up in a puddle of solution that contains heavy metals such as uranium. And this would result in images that have a dark background from the heavy metal, and then the particles themselves would be visible as bright areas in the images. For cryo-electron microscopy, the preparation process is quite different. Instead of a continuous support film, the support film will usually have small holes in it, and a thin layer of sample containing nanoparticles is suspended in these holes through a blotting process. And then the grid is rapidly frozen, resulting in a thin layer of vitreous ice. And this vitrification process spares the particles from freeze damage and results in particles that are preserved in their native hydrated state. Both the negative stain and cryo-electron microscopy have their place, but one important difference is that the contrast observed in cryo-electron microscopy images is directly attributable to the particles and their contents, as opposed to negative staining, where the contrast is a much more complex function of how the stain is interacting with the particles. Generally, negative stain is only revealing the exterior of the particles. For example, with these AAVs, you can see in the cryo-EM image below that you can directly detect DNA present within capsids or not. Whereas in the negative stain images above, the ability to detect the DNA depends on the ability of the stain to penetrate the capsid. In these LNPs, in cryo-EM, we can see that some of the blebs contain RNA and some of the blebs are empty. And in the negative stain image, it's not possible to even see the bleb structure due to the severe distortion of these fragile lipid-based structures. The grid preparation process is almost completely automated and handled by a robot. 3 microliters of the sample is placed onto a grid, and then that's placed into a chamber that's temperature and humidity controlled. And the robot blots the sample down to a very thin layer of liquid and then plunges the grid into liquid ethane that's being held near its freezing temperature. And then the sample is held at cryogenic temperature while it's loaded into the microscope. Once it's loaded, the grid is screened and areas are found that have ideal ice preservation, ice thickness, and vitrification. And then high magnification images are taken of the particles. And these images of the particles are 2D projection images. And these 2D images are routinely used by us to perform a number of characterization analyses, including particle sizing, fraction counting, looking for the presence of drug or impurities, particle aggregation, etc. And all of this analysis is usually being performed on two-dimensional projection images of the particles. But what if the characterization that you're trying to perform on your nanoparticles depends upon a three-dimensional assessment of the particle morphology? For example, if you're trying to disambiguate the interior of a particle from the exterior of the particle, or if you're trying to study the morphology of a particle that has a complex shape. I assume many of you are familiar with the technique of single particle analysis, which uses cryo-electron microscopy to generate high-resolution 3D structures of proteins and protein complexes, such as this apoferritin particle on the left. And the way this technique works is you collect many two-dimensional images of many copies of a single particle, and these images hopefully capture the particle in every possible orientation. And then there are methods for taking all these 2D images of this identical particle in these various orientations and combining them to compute a 3D volume of the particle. But for complex nanoparticles like these LNP particles on the right, that approach won't work because there's no single particle. Every particle is different, and trying to average a single two-dimensional view of many different particles would simply yield a blob. So tomography is a technique where we do collect 2D images of this single field of particles from many different orientations, and then we create a 3D volume of this field of particles. So this is a simple animation showing the process of electron tomography. You begin with a sample of nanoparticles, and they're frozen in a sheet of vitreous ice on a grid, and the grid's inserted into the electron microscope. And then an image is collected while the sample is being tilted every 2 or 3 degrees. And the total range of tilt goes from usually plus or minus 50 or 60 degrees. The sample itself is essentially an infinitely large sheet in the microscope, and it becomes thicker and thicker as it's tilted, and so it's impossible to collect all tilt angles up to 90 degrees. And this results in some missing information that leads to somewhat lower resolution in the Z dimension of the sample versus the XY dimension in the final 3D reconstructions. So then you take all the 2D projection images that are collected from the detector, and then you perform a reconstruction. And the reconstruction process begins by initially aligning all of the individual tilt images relative to each other to correct for any movement that occurred during the image collection process. And then there are a number of algorithms that take these aligned images. The most common one is called weighted back projection and generates a final computed three-dimensional volume of the nanoparticles. So now I'm going to walk you through the steps of doing tomography with a baculovirus sample. The first step is you begin with these unaligned tilt series images, and they look very noisy because you're trying to minimize the dose to the sample because it's still cryo-electron microscopy. You don't want to burn your sample or melt the sample, and you have to spread the dose out across all these tilt images. And so each individual image is quite noisy and difficult to see the details of the particles. And also at this stage, the images are still not aligned, so you can see all the movement that was happening during the data collection process. So now this is a line tilt series, and you can see that it's much smoother now. And in fact, your brain can already begin to process these images as they're moving, and you can begin to make out the three-dimensional shapes and relationships between the particles just by seeing how they change as the sample tilts. So now the tomographic reconstruction process has been run on the aligned tilt series, and we have a tomographic volume or a tomogram. So at this point, we can now dissect our particles slice by slice as we walk through the volume. And in this volume, we see there are a number of different types of particles that are present. You can see that there are baculovirus particles that have envelope with a certain number of spikes. This particle has a lot of spikes all around its envelope. This particle has patchy areas of spikes with other areas lacking spikes. Here's another very long virus particle that is obliquely oriented through the volume, but you can see that there's again a lipid envelope and a nucleocapsid, and most of the envelope is devoid of spikes. You also see there's liposomal particles, small round particles that have lots of spikes. And you can also see that the nucleocapsids themselves, some of them are empty and some of them are dense. You can see the end caps on the nucleocapsids. And then you can also see that there's different types of debris or contamination in these samples. There's this dark flocculent looking debris that's interspersed throughout the samples. And then other areas you have these sort of lumpy cauliflower shaped particles that are present in the sample. So one way that you can begin to appreciate the three-dimensional nature of this data is to create volume renderings of the data sets. And this is similar to what you typically be doing maybe with confocal data, for example, because we have dark particles on an otherwise bright background. You can have some success with this approach. And for example, in this volume, you can see that there are some particles that have these coronas of bright spikes on their surface. And then you have other particles that are naked and lack the spikes. And this is a pretty easy way to start to see the three-dimensional structures that you have in your data. In order to extract the most information from these volumes, you need to perform object segmentation. And this is a process where you go through all of these slices and you draw contours in a somewhat semi-automatic way usually. And this allows you to create three-dimensional objects that represent the particles in your sample. And once you have these segmented objects, you can now measure their volume, their surface area, you can measure distances between them, and you can start getting real quantitative data out of these three-dimensional data sets. And so here you can see that the viruses have been segmented to show the nucleocapsids in magenta and the envelopes in green. I'm going to move on now to a different type of virus, lentivirus. Lentivirus samples can be challenging samples to look at in the cryo-electron microscope. You can see a lot of interesting details in two-dimensional images for sure. For example, in these 2D images, you can see that these particles clearly resemble lentivirus particles. And you can see that there are spikes on the surface of the particles. You can see these are probably mature virus particles. And once in a while, you can even start to make out what appears to be a capsid in the particles. But generally, the density of the particles makes it very challenging to convince yourself for sure that there's capsids inside the envelope. And this is something that's known to vary in lentivirus samples, the presence of capsids. And so if you want to confirm for sure that there are capsids intact, that's hard. And then furthermore, in a lot of these lentivirus samples, there is often a lot of debris along with the virus particles themselves, lipid debris, protein debris, liposomes, and all this debris can begin to obscure and make it difficult to make out the details of the virus particles themselves. And this is where tomography can be useful for looking at lentivirus. So I'm going to show you a tomographic reconstruction of a field of lentiviral particles. You can see that there are a number of particles of various shapes and sizes present. And as you zoom in on a particle, you can see a very clear conical, cylindrical capsid structure. You zoom in on a couple of other examples. Here you can see a capsid. Here you can see another capsid. Here's a capsid structure right here. And you can also see on the surface of the particles very clearly individual spike proteins that are being displayed. And so we did this across a set of a number of tomograms and looked at over 100 virus particles, and we were able to show that 71% of the particles contained a clear capsid structure as far as we could tell from this tomographic process. And before I move on, one more point I thought I would discuss here is subtomogram averaging. Perhaps you've heard of this. This is not a technique that NIS has offered historically, and I'm not going to go into depth in this talk, but I did want to point out that there is a process where if you have a situation where you have perhaps protein that's being displayed on the surface of a viral vector, for example, or maybe even a LNP, any kind of particle, then you can use a process that's a combination of tomography and the single particle averaging approach that I was discussing in the earlier slide, where we were looking at apoferritin. And you can collect a number of tomograms, maybe dozens of tomograms, where you have hundreds of particles, and that ends up giving you thousands and thousands of these spike proteins across that population. And then you can go through and extract each one of these proteins, and hopefully they're caught in many different orientations, and then you can use this single particle averaging technique to generate high resolution 3D structures for the proteins. And you could even, if you have enough data, potentially begin to see differences among the population. For example, perhaps you have some prefusion and some postfusion conformations, or maybe you're expressing two different proteins on the surface of your vector, and you want to confirm that both are being displayed. So this is a relatively intensive process, both in terms of the data collection and the data analysis. And again, it's not something that we have offered up till now, but it is something that if we have clients, if we have people who come to us and tell us that it's something they'd be interested in doing, we'd certainly be very interested in working with you to potentially do subtomogram averaging. Now I'm going to change gears a bit and talk about lipid nanoparticles. This is a sample that was supposed to contain some lipid nanoparticles, or LNPs, with a surface modification to express a protein that would allow for targeted delivery. And as you can see from these traditional cryo-EM images, there was a lot of protein in the samples. Unfortunately, there's so much protein throughout the images that it's very difficult to discern whether or not any of it is perhaps specifically associated with the surface of the particles. And you can also see that some of the particles themselves have a very strange profile in these 2D images. In electron tomograms of the sample, you can see that there is a lot of protein throughout the sample and also across the surface of the sample, but it appears to be randomly distributed throughout the sample and not specifically associated with the surface of the particles. If you look along the surfaces of all the particles, you do not consistently see any protein there. And looking at a number of tomograms and many different particles, it becomes apparent that the surface modification of these particles with the protein appears to have been unsuccessful. You can also see in these volumes that a lot of the particles have a very complex morphology. There's sometimes blebs within blebs, even particles inside of particles. And these are the sorts of relationships that would be very difficult to study in a two-dimensional image. But in 3D electron tomograms, you can segment out the exterior of each particle and also the internal features of the particle. And then you can study the relationships between these. And this allows you to study a number of different particle types. For example, let's say you have particles that have a porosity to them and you want to study the porous structure of a particle. Or perhaps you have particles that have a core shell structure and you want to study the relationship between these internal structures. This is something that electron tomography would be particularly powerful for doing. The next example is another lipid nanoparticle formulation that was also supposed to have a surface modification, this time with antibodies for targeted delivery. When you look at 2D cryo-EM images of these particles, now you can see, first of all, it's a much cleaner sample. There's no aggregates of protein throughout the sample. And you do see that there are some small densities near the surface of some of the particles, these little faint specks around some of the particles, although most of the particles appear to not have any or very few of these specks. And so it's difficult to tell whether or not maybe there's a lot of labeling here and it's hard to see due to the low contrast and the noise that's present in the images. Or perhaps there is some labeling, but there's just not a lot of particles that are labeled and there's not a lot of labels per particle. When you look at these particles in electron tomograms, you see a lot of interesting things. First of all, that some of these particles have internal densities to them. There's dots in some of these particles, there's squiggly lines, there's multilamellar looking structures. And this is a phenomenon that has been studied using electron tomography by groups that have published papers. These appear to be associated with lipid RNA interactions. And this is a very interesting observation that we see in a number of LNP formulations. It's also possible to see that a number of the particles have triangular morphologies, lentil-shaped morphologies. They're not all round spherical particles. But to get back to what I was discussing before, most of these particles appear to have no protein on the surface, but there are a few here in the middle of this sample where you see that there are proteins on the surface of the particles, and they even appear to have what you would expect to have antibody morphology. And there's also particles that appear to be maybe aggregating, tethered to each other through some protein interactions where the proteins are sticking to each other. Based on these images from the middle of the section, it looks like there's some protein, but most of the particles look like they have little or no protein on their surface. And then you get to the air-water interface, the very surface of the sample, and all of a sudden you see there is a ton of protein in the sample. Almost every single particle has this halo, this corona of protein completely surrounding it, but it's all stuck at the air-water interface. This is a phenomenon that commonly occurs with proteins. It's something that people doing single particle analysis struggle with commonly. And if you actually map the distribution of protein that's associated with each particle at the surface, and then you go down into the volume further to where you see the maximum diameter of each particle, you can clearly see why it would be hard to see the proteins if you're looking down on these particles from 2D. And that's because the particles all have a structure that looks something like this, where you have a halo or a corona protein at the surface, and maybe some also at the other surface, at the other air-water interface, and the majority of the particle has no protein associated with it. And so if you were looking down from above, it would be hard to see any protein on this particle. So this is an artifact of the sample preparation process, I'm sure. If these particles are floating around in solution, I would think that most of this protein is diffusing along the surface of this LNP. But because we did electron tomography, we're able to see that these particles are actually very robustly labeled. Almost every particle has protein associated with it, and appears to be quite a good amount of labeling. One more final example, and this is another lipid nanoparticle formulation. In this sample, we were seeing these strange round particles. It was hard to tell if they were real particles or if perhaps these are very thin sheets of material that are right at the surface of the sample, which would explain why they have such a low density compared to the classical, typical LNPs that are throughout the sample. Very hard to answer that question without looking at the sample in 3D. And then I'll also briefly show an attempt to analyze the structure of the blebs and the structure of some of these texture patterns that are seen in the LNP particles. When you look at this sample in tomographic volumes, you find that there are, in fact, actually two separate populations of particles. There's the classic, typical LNP particles, but then there's also this subpopulation of particles that represent those very faint particles we saw in the 2D images, and they appear to be disintegrating or falling apart at the air-water interface. And so when you segment out all of these particles and you look at them in 3D, you realize that these particles are actually hemispheres, and half the particle has fallen apart at the air-water interface. When you zoom in and look at these two different populations at higher magnification, the typical LNP particles generally have a lipid bilayer at their surface, whereas these particles that are sensitive to the air-water interface, they consistently lack a lipid bilayer. They also appear to lack blebs as far as I can tell. And so this is interesting in light of the sample we looked at previously, where you have the antibodies actually anchoring the LNPs at the air-water interface, and we didn't see any examples of particles that were falling apart or hemisphere-type particles like we see here. So there's something about the way that the surface of these particles is formed that's making them very sensitive to the air-water interface. So another thing we did with this sample is we segmented out almost 100 particles and all of their blebs and looked at the volume analysis of the bleb structure. We found a few examples of particles. Out of these, less than 5% of the particles had blebs at one pole of the particle, which would have made it hard to see these blebs in a 2D perspective, but it was relatively rare that this sort of orientation was found. And so when you look across the full population of all the particles that we segmented out, we were able to break down the particles into categories in terms of the number of blebs that they have, and we were able to calculate for the particles that did have blebs the average volume fraction of the blebs for this formulation was about 5%, and the other 95% of the particle would be solid lipid phase. So one last thing that we looked at with these particles were these dense structures that were observed within the lipid compartment. And they weren't observed in every particle. Some particles had none that we could tell, or very few. But there was a sizable subpopulation of particles that had these very prominent densities just along the subsurface of the particle. And it was possible to, based on pixel density, automatically segment out some of these structures and reveal these thin threads that are running along the subsurface of the particle. And the average diameter of these threads was 2.5 nanometers with an 8.2 nanometer spacing from thread to thread. And so, again, these are believed to be associations of the RNA with the lipid phase. And for this case, at least for this particular formulation, there's little evidence for much texture within the bulk of the lipid phase, but these consistent threads along the very subsurface of the particle. But that would differ from formulation to formulation for LNP samples. So to quickly wrap it up, I hope I've managed to convey what an interesting technique electron tomography is and the types of unique insights that you can obtain through tomography. It's definitely not a technique that you would use on a regular basis for every sample. It's lower throughput than conventional cryo-electron microscopy, and the data analysis does take a bit of effort to actually extract quantitative 3D data out of these volumes. But then again, sometimes just taking a quick look at a sample with electron tomography can immediately give you some idea of what's happening in your sample. For example, those particles that were falling apart at the air-water interface. So with that, I think we're ready to take any questions that may have come in.
Thank you, Eric, for that great presentation. Before we move to the Q&A session, I wanted to provide our email address, which is info@nanoimagingservices.com. So please do reach out if you'd like to meet to discuss a potential project or learn more about our capabilities in that setting. And in addition to our standard cryo-TEM services, including tomography, I did want to mention that NIS is the only GXP compliant cryo-EM lab in North America, and we'd also be happy to chat with you about supporting your regulatory submissions. So we're now going to the live Q&A portion of the webinar. So if you have a question, you can submit it in the Q&A box in the toolbar. So the first question is about resolution. So we have someone who is bringing up a project that they're working on, and they commented on the resolution of the data. So Eric, maybe you can compare the resolution that you would get with this technique tomography with maybe something generated by single particle for high resolution work.
Sure. So these tomograms, especially as you're seeing them in this presentation, they don't have nearly the resolution that you would see from coming out of single particle analysis, the high resolution structures that come out of that process. The tomographic volumes in the XY plane, they typically will have the same resolution that you'd have in your images that you get off of a microscope, a couple nanometer resolution, let's say. In the Z direction, it's going to be maybe 1.5 to 2 times worse because of that missing information problem I discussed during the tilt series collection. Whereas obviously these protein structures that come out of the single particle analysis, they have a couple, 2 or 3 angstrom resolution, 10 times better. But you can, again, potentially perform subtonogram averaging to pull that kind of resolution out of these data sets. The volumes you were seeing in this presentation, I put all of them through some denoising algorithms work to try and clean them up, because the data as it comes out of the reconstruction process, it's initially a bit noisy. Yes, especially if you're just trying to manually segment them and just see things by eye, you perform some denoising, and the trade-off is a bit of resolution that way.
Good point. Our second question is for the samples for LNP projects. So for LNP specific samples, the particles are often clumping together. So what are the best practices to keep them nicely separated as they appear in your beautiful examples that you showed today?
Part of that is probably due to a peg layer, which is invisible in the electron microscope. It's there, but you can't see it. And so one of the ways that we infer that it must be there is that when you see particles in these, like a dense field of particles, for example, the ones that have the antibody labeling that we saw in I think the second LNP example, none of the particles were touching each other because there's a peg layer there stopping them from touching each other. So part of it is just the nature of the particles themselves. If they are clumping together and you have a peg layer, I guess that might be problematic. One of the things we can assess is aggregation in samples, and if something is happening.
Right.
Sometimes If, for example, you saw that it looked like this protein tag that was put on the surface of the particles may have been causing them to clump together somewhat. And now that may even be partly why they were forming these arrays in the images to some extent. That
Right.
may have been helping to contribute to that. So it comes down to your concentration also too. If you have just a lot of particles, they have nowhere to go. You can try some dilution and things like that to
Right.
optimize the grid prep itself.
This segues into our next question is about the antibody conjugated LNP. So with the it shows the majority that of the antibodies are on the top and on the bottom. So is this real or is this an artifact due to the sample prep?
I think it's an artifact due to the sample prep. It's a well-known artifact that a lot of proteins, they'll be stuck at the air-water interface.
Mhm.
The grid preparation process is pretty quick, but it does take
Mhm.
seconds, and that's plenty of
Right.
time for the protein to find its way to the air-water interface and get stuck there if that's where it likes to be due to the just to the properties of the protein. Not every protein does this, but there's some that do. For people doing spa type work, the single particle analysis, they have all kinds of tricks like adding detergents and things to their samples, but that's something especially for an LNP sample, that would be harder perhaps to do. So I think that's a sample preparation artifact, but nevertheless, and that's a scenario where we still learn something important about the sample that we weren't able to see in the 2D images, and tomography at least allowed us to see that the antibody labeling was working for these particles. They were
So switching gears more to the data collection, how many volumes do you typically collect per sample? And how many particles do you usually see for this analysis?
That's adaptable. It really depends on the scope of the study, what you're trying to learn. As I said towards the end, you can learn a lot just sometimes by looking at a couple of tomograms and get an idea of what your particle structure is. And that's something we routinely do for clients. We delivered a couple of tomograms and some
Mhm.
movies showing what they look like in 3D. And then
Right.
if you have a greater need, more data, you want to really do analysis and get a lot more particles, then we can maybe spend a whole day on the microscope and get probably closer to 50 tomograms of
Mm.
your sample. And theoretically, again, if clients come to us and want to do subtomogram averaging, that could be 2 or 3 days on the microscope, getting enough tomograms to have enough particles to be able to do a subtomogram averaging project. So it's adaptable, it's customizable to what you need.
So how much sample do we typically ask for this type of analysis? And is it different than our standard, I'll say the classic imaging that we do?
And we usually ask for at least 50 microliters of a sample. It takes 3 microliters to make a grid, so that
Mhm.
is generally enough for us to do some grid optimization and get to a point where we have grids that are good for doing the imaging. And 50 microliters is usually enough.
Okay. Moving back to the data, so the data analysis, does NIS perform the 3D quantitative analysis for this data?
Not usually. For perhaps down the road, the subtomogram averaging, we have obviously experts who do a lot of this sort of a high resolution structure determination. I imagine that we would be able to help out with that process. For a lot of the analysis that you saw in this talk, where I was segmenting out the LNPs or segmenting out the viral particles, I was doing all of that using free open source software that anybody can download and use. And I would say it's really on the client. If that's what they want from the data, then they'll have to do that themselves. But
A lot of work.
it's not so bad because the software it's been developed to minimize the pain.
It's come a long
In a
way for
few
sure.
hours, you can have a lot of particles segmented.
Wonderful. So how can you tell if the vitrification process was successful?
Well, that's pretty clear from looking at the images, just in the 2D images as you're screening the grid. Crystalline ice, you'll have diffraction patterns in your images. You'll see
Mhm.
these black and white blotches in your images. And so it's readily apparent if you don't have a good amorphous vitrified ice. And then in terms of ice thickness, fortunately for tomography, it's not so important. You don't want your sample getting too thick as you're tilting up to the higher angles.
Right.
But if your sample's a bit thicker, that can actually be nice. You just have more particles there, bigger particles that you can look at in your final 3D volume. Once you're slicing through your volume in these computed slices, that thicker ice is not such a big problem in terms of contrast and being able to see details.
Going back to comparing tomography versus our single particle workflow, could you describe any of the key advantages of tomography over our high resolution 3DR workflow and when you would choose one over the other?
Whenever you have particles there, every particle is like its own little snowflake and they're all different, then you can't use single particle
Right.
averaging approach. And so for any kind of complex nanoparticle like LNPs or viruses, tomography is really the only way to go to get 3D information out of those sorts of particles. If you have you have proteins, then obviously a single particle analysis would be the way to get 3D structures and high resolution data out of that. And then subtoner imaging is the hybrid approach where if you have some consistent little particle that's in your tomograms, people usually apply it to things like finding ribosomes or finding other protein complexes that are inside their cells, and they'll go through and they'll find them, and then they can ultimately pull out very high resolution structures in the angstrom
Mhm.
resolution range from these tomographic volumes.
All right, so we have time for one more question. Someone's asking, what are the common pitfalls that you see in the sample prep for cryo-electron tomography? And do you have any recommendations for addressing them?
The sample prep, sucrose is a problem. Any cryoprotectants can reduce the contrast, and so once you get up, if we receive samples that have a lot of cryoprotectant, let's say 10% sucrose or trehalose, then that can interfere with, first of all, just getting good freezing. If it starts to get to that point, you can sometimes even interfere with the freezing. But generally, definitely can interfere with the contrast. And so sometimes we'll dilute samples to reduce the presence of cryoprotectants. Hopefully, there's enough particles in there to allow for that. And then, yeah, and then just finding the right concentration of particles in your sample so that you have enough samples that you have enough particles there so that when you do a tomogram, hopefully you have a field of many particles there that you'll be able to assess your sample. And we can dilute some, but if you don't have enough particles there, then that would be problematic. So you want to have samples generally where a nanoparticle will be maybe in the E12 range per
Okay.
milliliter, particles per milliliter, and that should give enough particles per field of view. Hopefully we can find some areas to collect the tilt series and give you more than just one or two particles in a tomogram.
Right.
And that can also be problematic because in traditional tomography, sometimes you can add little gold nanoparticles to act as fiducial markers, and they help with the tilt alignment process. We generally try to avoid, most of our clients want us to avoid that. Frankly, they don't want to be adding anything to their sample. But that means that you do need to have enough of your actual nanoparticles in there that they can act as the fiducial markers themselves.
Oh,
And so you have enough information in the image to do the tilt alignment process.
gotcha. All right, thank you. And with that, this concludes our webinar on cryo-electron tomography. I'd like to give a big thank you to Eric for his great presentation and for answering questions, and to all of you that attended today and contributing. If we weren't able to get to your question today, we will send an email reply. But also, if there's anything you can think of after the webinar, please reach out via email to info@nanoimagingservices.com. And as a reminder, a copy of this recording for the webinar will be sent to all of those that registered. So with that, another big thank you to Eric, and we'll say goodbye. And thank you to everyone for attending.